To understand why the finding matters, you first have to understand how unusual it is for a modern population to retain a clear and direct genetic connection to the people who lived in the same geographic region 5, 6, or even 7,000 years ago. The history of the world, as ancient DNA has revealed over the past 2 decades, is overwhelmingly a history of replacement. Populations don't typically persist in place across multiple millennia. They get absorbed, displaced, overwritten. The people who built Stonehenge were genetically replaced by incoming Bronze Age farmers from continental Europe. The Hunter-N, a few generations. The ancient Indus Valley civilization, which at its height housed more people than ancient Egypt and Mesopotamia combined, collapsed so thoroughly that the genetic identity of its builders was essentially dissolved into the populations that followed. The ancient Egyptians, whose genetic profile during the pyramid building era was predominantly connected to Neolithic, Near Eastern, and North African farmers, with a significant Mesopotamian component, were progressively replaced over the following millennia as sub-Saharan African ancestry increased substantially in the region until the genetic distance between the Mummy-era population and modern Egyptians became measurable and significant. This is the rule of ancient genetic history. Populations replace populations. Civilizations end, and the people who built them either disappear, migrate, or are absorbed into something genetically new. And then there's the Yellow River Basin of northern China. Researchers studying ancient DNA from Chinese populations across multiple archaeological periods have demonstrated that the genetic history of China's Central Plains began with Neolithic millet farmers of the Yangtze culture, who constituted the region's foundational population. Studies of individuals from the Qing dynasty have confirmed that they are direct ancestors of the modern Han population in Henan, completing an unbroken multimillennial genetic lineage. The findings demonstrate the stability of the genetic profile of the Central Plains population, a stability that has persisted for millennia and remained profoundly unaffected by major historical upheaval. Read that carefully. An unbroken genetic lineage, from Neolithic millet farmers to Qing dynasty individuals to modern Han. Chinese, not a similarity. Not a partial match, a direct ancestral line, confirmed by genome-wide data, stretching across more than 5,000 years of Chinese history without a fundamental replacement event. The Yangtze culture was a Neolithic culture that existed extensively along the Middle Yellow River and surrounding regions in China from approximately 5,000 to 2900 BCE. It is the largest, longest-lasting, and most influential archaeological culture in prehistoric China and held great significance in the early development of Chinese civilization. Its sphere of influence spanned the Hetao area in the north, bound by the Dawenku culture of present-day Shandong province in the east, and the Majiayao culture in the northwest. The Yangtze people were millet farmers. They lived in semi-permanent villages, built pottery, developed early symbolic systems, and laid what genetic research now confirms is the biological foundation of the largest ethnic group on Earth. Their descendants, genetically traceable, genomically confirmed, are alive today, walking around, eating meals, sending messages on smartphones, living in cities that sit on the same river plains where their Neolithic ancestors planted millet in the Yellow River, Loess, more than 7,000 years ago. Recent ancient DNA studies highlighted that the Yangtze culture was one of the most important biological roots of East Asians. The populations in the Central Plains exhibit long-term genetic stability and continuity. The scientists who study this don't use words like stability lightly. In a field where the standard story of any given region usually involves multiple waves of replacement and mixture events that rewrite the genetic record and populations turning over every few thousand years, the word stability applied to a 5,000-year genomic record is genuinely extraordinary. It means that whatever forces produced population replacement everywhere else in the ancient world, the migration of steppe pastoralists, the expansion of agricultural frontiers, the collapse and replacement of civilizations, did not do to the Chinese core population what they did to the genetic lineages of most of the rest of the world. But then came Shimao, and everything became more complex and more astonishing at the same time. Shimao is a site in northern Shanxi province in China that, when archaeologists first fully appreciated its scale, they struggled to fit into any conventional narrative of Chinese prehistory. Shimao City, dating to around 2300 to 1800 BCE, is a premier state-level Neolithic fortified settlement in Shanxi. The stone-walled site encompasses roughly 4 square kilometers and can be divided into outer and inner enclosures, showing features typical of state-level societies: craft production, large fortifications, and high social stratification with abundant forms of human sacrifice. With a strict hierarchical polity and a unique culture of human sacrifice, more than 80 human skulls were found buried under its east gate. Shimao serves as an excellent illustration of the roles of family and ancestry in the structuring of political and social relationships in early state-level human societies. 80 human skulls under the gate of a city. This is not a footnote. This is one of the most concentrated collections of sacrificial human remains from the prehistoric world, and it sat in northern China, connected to a civilization that most of the outside world had never heard of, waiting for DNA analysis to finally explain who these people were and where they came from. That analysis came in November 2025, when a team led by Professor Qiaomei Fu of the Institute of Vertebrate Paleontology and Paleoanthropology of the Chinese Academy of Sciences published what became an immediate landmark in ancient DNA research. The team spent 13 years carrying out large-scale, high-resolution nuclear genome analysis on 169 ancient human samples, collected from Shimao and surrounding sites, as well as the southern Shanxi region. Analyses of ancient DNA from individuals who inhabited the site reveal genetic links to southern settlements, while also providing evidence of male mass burials associated with human sacrifice. 13 years. This was not a quick sequencing project. This was a sustained scientific excavation of the most intimate evidence of a civilization's identity, its DNA, conducted with the painstaking care that only makes sense when the researchers understand they are sitting on something historically significant, and the results confirmed with a level of genomic detail that previous generations of scientists could never have achieved, that the people of Shimao were not a foreign population that had arrived from somewhere else. Genetic analysis shows that the Shimao people can trace the bulk of their ancestry to local Yangtze culture farmers who tilled the Loess Plateau more than a millennium earlier, underscoring an unbroken chain of regional genetic continuity. The city's founders were not steppe invaders. They were not migrants from Central Asia or from the West. They were the genetic descendants of the people who had been farming millet in the same general region since the Neolithic period, descendants who had built, over more than 1,000 years, the organizational capacity and social complexity to construct a 4-square-kilometer stone-walled city, maintain a ruling class power structure that spanned at least 4 documented generations, and conduct ritual human sacrifice on a scale that had no parallel in China at that time. Analysis of tombs from elite burial areas revealed a society organized primarily along patrilineal lines. High-status male tomb owners were central to the kinship networks, with their wives genetically traced to different external biological families. The ruling class brought their wives in from outside the core genetic group. They maintained biological diversity in the female line, while consolidating patrilineal power through the male line. This is a pattern seen in dominant hierarchical societies across human history, but what makes its appearance at Shimao remarkable is that it was happening 4,000 years ago in a society previously considered too early for this kind of sophisticated social stratification. Victims from a skull pit were predominantly male, while those sacrificed to accompany elite individuals in tombs were almost exclusively female. The sacrificed individuals showed no close biological kinship to the tomb owners, but some sacrificed females were found to be biologically related to each other, suggesting that specific families or communities might have been selected for sacrificial rites. Specific families selected for sacrifice. The genetic data reveals not just who these people were in terms of ancestry, but how the killing was organized. Families contributed victims. Communities were systematically harvested for the ritual requirements of a ruling class that had turned biological lineage into the currency of both power and death. The science here does something that archaeology alone cannot do. It reveals the human cost of ancient statecraft written not in text, but in the genome itself. But before Shimao, before even the Yangtze culture farmers who built it, the genetic story of the Chinese people reaches back into a deeper and stranger past than most people realize. The ancestors of the Chinese, like all modern humans outside of Africa, carry within their genomes the faint biological fingerprints of encounters with extinct human relatives. And in East Asia, this ancient inheritance takes a form that is found almost nowhere else on Earth. The hypoxia pathway gene EPAS1 in Tibetan individuals has an unusual haplotype structure that probably resulted from introgression of DNA from Denisovan or Denisovan-related individuals into humans. This haplotype is only found in Denisovans and in Tibetans, and at very low frequency among Han Chinese. The Denisovans were a separate branch of the human family tree, known from just a handful of physical fossils, a finger bone, some teeth, a jawbone discovered on the Tibetan plateau, but whose genetic legacy has been traced in modern human populations across East Asia and into the Pacific. Scientists suspect that modern humans had sex with Denisovans in Asia somewhere between 30,000 and 40,000 years ago. They inherited the Denisovan version of EPAS1, which lingered in populations at very low frequencies. The carriers fared better at higher altitudes, and their descendants colonized the Tibetan. Plateau. This explains why researchers find the Denisovan E-PES1 in the vast majority of Tibetans, but also in a couple of Han Chinese people living outside of Tibet. The altitude gene that allows modern Tibetans to live and work and give birth at elevations where most of the world's population would suffer chronic oxygen deprivation is not, genetically speaking, a human invention. It was borrowed from a species that no longer exists. It was preserved in the gene pool because it worked. And it sits today in the genomes of millions of people across the Tibetan Plateau and its surrounding regions, a silent record of an encounter between two branches of the human family that happened 40,000 years ago in a mountain range in Asia. Modern Han Chinese, the genetic research confirms, carry traces of both Neanderthal and Denisovan ancestry, as do all non-African human populations. But the specific Denisovan contribution to the East Asian gene pool has features that set it apart from the Denisovan admixture found in populations further south and east. The geographic range and population structure of Denisovans are largely unknown, although genetic studies of modern humans have given some tantalizing, if incongruous, hints. A 160,000-year-old mandible from Baixia Karst Cave in Gansu, China, has been characterized as likely Denisovan through ancient protein sequencing, and sediment DNA from the cave provided the first genetic proof of Den unequivocally extending the known range of Denisovans. Denisovans were in China for at least 160,000 years before modern humans arrived. They were not passing through. They were inhabitants of the same land, adapted to the same environment, using the same caves. And when modern humans finally arrived and encountered them, the two populations did what humans so often do when they meet. They had children together. Those children's descendants became, in part, the people of China. Now, return to the central question. When scientists compared Chinese ancient and modern DNA to every other civilization in the genetic record, who matched? The answer requires understanding what match means in this context. It does not mean that ancient Egyptians or Mesopotamians were genetically identical to ancient Chinese people. They were not. It does not mean that there was direct contact or migration between early Chinese civilization and any specific near Eastern or Mediterranean society at the genetic level. The ancient DNA evidence does not support that kind of claim. What it means is something more profound and more specific. When researchers looked at the degree of genetic continuity between an ancient population and its modern descendants in the same geographic region compared across every major ancient civilization in the genomic record, only one civilization showed an unbroken biological thread connecting its Neolithic founders to its modern population: the civilization of the Yellow River Basin, which grew into the culture and people we call Chinese. Research published in Molecular Biology and Evolution found that the lower reaches of the Yangtze and Pearl rivers, as well as the Yellow River, gave rise to genetically separate groups about 10,000 years ago. Those ancestors then mingled to become the largest ethnic group in the world today. The history of Han Chinese is more sophisticated than thought, the lead researcher concluded. 10,000 years. The genetic roots of Han Chinese identity reach back into the post-Ice Age period, when the Yellow River and Yangtze river basins were being settled by distinct but related populations of early East Asians who had made their way through Southeast Asia on the long journey north and east from the original African exodus, Africa, and immigrated into East Asia via a southern route approximately 50,000 years ago. Following the end of the last glacial maximum approximately 12,000 years ago, rice and millet were domesticated in the south and north of East Asia, respectively, which allowed human populations to expand and linguistic families and ethnic groups to develop. In central China, pronounced population admixture occurred and accelerated over time, which subsequently formed the Han Chinese population and eventually the Chinese civilization. 2 rivers, 2 crops, 2 foundational genetic lineages, northern millet farmers along the Yellow River and southern rice farmers along the Yangtze, that gradually came together over thousands of years, mixing and merging and sharing genetic material until they produced the population that built the first Chinese cities, wrote the first Chinese characters, performed the first recorded Chinese rituals, and established a civilization that, unlike almost every other ancient civilization on Earth, never fundamentally stopped. Northern China harbored the world's earliest complex societies based on millet farming in 2 major centers in the Yellow River and West Liao River basins. The Yellow River populations show a monotonic increase over time in their genetic affinity with present-day Southern Chinese and Southeast Asians, suggesting ongoing genetic mixing between northern millet farmers and southern populations across the full span of Chinese prehistory. This gradual genetic mixing is itself one of the defining characteristics of the Chinese genetic story. Unlike the dramatic population replacement events seen in Europe and West Asia, events where one population essentially overwrites another in a geological instant, the formation of Han Chinese identity was a slow, continuous process of regional admixture between populations that were already genetically related, living in proximity, and interacting across agricultural and cultural boundaries. From the Longshan culture until the Iron Age, populations in the Central Plain show strong genetic continuity with limited or no genetic contribution from the surrounding regions. Through the Jia, through the Shang, through the Zhou, through the Han dynasty that gave the ethnic group its name, through the Tang and the Song and the Ming and the Qing, the genetic core of the Central Plains population maintained a remarkable stability that has no clear parallel among the ancient civilizations of the Western world. Scientists from Peking University have uncovered new genetic evidence that sheds light on how prehistoric people in China interacted, migrated, and built their communities. The research reveals the first direct genetic proof of a patrilineal social system in Neolithic China. This research provides new insights into how early Chinese communities adapted to environmental and cultural changes, as well as how their family systems were organized. The story of Chinese civilization begins along two great rivers: the Yellow River, known for its millet farming cultures, and the Yangtze River, home to early rice agriculture. The patrilineal system revealed in the Neolithic DNA, the pattern of men staying local while women married in from outside, created a specific kind of genetic structure in ancient Chinese communities. Men passed their Y chromosomes down through stable patrilineal lines. Women moved between communities, diversifying the maternal genetic record. This social architecture, confirmed in the bones of people who died thousands of years before writing was invented, is the biological skeleton of a kinship system that persisted in Chinese culture through recorded history. The family structures that later Confucian philosophy would codify and sanctify were already being written into the genetic fabric of Chinese communities during the Neolithic period, centuries before Confucius was born. And the genetic structure built in that ancient period proved remarkably resistant to replacement, even when the forces that overwhelmed other ancient populations came knocking. The Yangtze Steppe expansion, which rewrote the genetics of Bronze Age Europe in a single generation, reached the northwestern frontiers of China, but did not fundamentally alter the core genetic profile of Yellow River farmers. The Mongolian expansion of the 13th century CE, which physically conquered a larger contiguous territory than any empire in history, left surprisingly little genetic signature in the Han Chinese core population, compared to the demographic disruption it created. Conquerors came and went. Dynasties rose and fell, borders were redrawn across the map of East Asia again and again across 4,000 years of recorded history. And the genetic profile of the people farming the Yellow River Loess, building cities on the Central Plains, maintaining ancestral lineages through patrilineal kinship systems, that profile endured. This is the finding that no other ancient civilization produced when scientists ran the comparisons. The ancient Sumerians of Mesopotamia have no direct genetic descendants in the modern population of Iraq. The population of the region has been fundamentally transformed by successive waves of migration and replacement across 4,000 years. The first genome, sequenced from an ancient Egyptian, revealed that 80% of his genetic material came from ancient people in North Africa, while 20% traced back to people in West Asia and the Mesopotamia region, a genetic profile significantly different from that of modern Egyptians, who carry substantially more sub-Saharan African ancestry. The builders of the pyramids and their genetic descendants have diverged. The Indus Valley Civilization, one of the most sophisticated urban societies of the ancient world, left genetic traces in modern South Asians, but not as a continuous and unbroken lineage. The genetic contribution of Harappan people to modern Indians is diluted and mixed with the subsequent Bronze Age arrival of steppe-related ancestry that transformed the genetic composition of the subcontinent. Even the ancient Greeks, whose cultural legacy is credited as the foundation of Western civilization, were genetically different from modern Greeks in ways that reflect multiple subsequent population movements through the Aegean. Only the Yellow River population maintained the kind of direct, genomically confirmed, generationally unbroken biological continuity between ancient founders and modern descendants that made the comparison across civilizations produce a singular result. A groundbreaking genetic study of China's Shimao ruins has provided definitive evidence that the core population of the Shimao culture descended from late Yangshao communities in northern Shanxi province, reinforcing the theory of China's civilization having evolved continuously on its own soil. Continuously on its own soil. These are words that carry a specific and verifiable scientific meaning when they appear in a peer-reviewed paper published in Nature. They are not poetic claims about cultural heritage or national identity. They are statements about what the DNA actually shows, that the people who planted millet along the Yellow River in the 5th millennium BCE and the people who live along that same river today share a direct and measurable biological inheritance that no other pairing of ancient and modern population in any other civilization has been shown to match. The implications of this radiate outward in multiple directions simultaneously, They raise questions about what made the Chinese population resistant to the kind of catastrophic replacement events that remade the genetics of every other ancient civilization. They suggest that the geography of the region, the natural barriers of the Gobi Desert, the Tibetan Plateau, the mountain ranges of the west and north, may have created a degree of demographic protection that other ancient civilizations lacked. They raise the possibility that the social structures of ancient Chinese society, the patrilineal kinship networks, the agricultural communities tied to specific river systems, the deep investment in ancestral continuity that became the philosophical backbone of East Asian culture, were themselves a form of biological protection, a social technology that preserved genetic lineages across the chaos of history. And they force a reckoning with a question that the DNA alone cannot answer, but that it makes impossible to ignore. What does it mean for a people to exist continuously in the same place, maintaining recognizable biological connections across 5, 6, 7,000 years of human time? Most of the world's populations cannot answer that question from their own genetic record. The ancient Mesopotamians cannot. The ancient Egyptians cannot. The ancient Harappans cannot. The Bronze Age Europeans certainly cannot. But the descendants of the Yellow River millet farmers, the modern Han Chinese people, numbering over 1 billion individuals, carry within their cells the molecular evidence of an extraordinary persistence, a civilization that faced every disruption the ancient world could throw at it, and, unlike every other ancient civilization on the genetic record, never stopped being itself.